This is Brenda Wingard-Haynes, and this is Biochem 208, and we are working our way through Unit 6. I am at home, and I've got my dogs in the background keeping us company, so I guess we're just going to have to embrace their presence during this presentation. We're talking about enzymes and factors that affect their reaction rate, and we're seeing that temperature and acid are strong destabilizers of enzymes. And where we left off was with the discussion of blood acidosis. Remember that acid is a powerful destabilizer of hydrogen bonds, so it makes a lot of sense that very quickly we can start having a variety of problems, including loss of consciousness, coma, and heart problems. All right, now, what can happen if there's a metabolic problem or some sort of genetic problem in which not enough enzyme is made? Well, we've talked about this before, but remember in Unit 4 when we talked about lactose and the fact that it's got this beta bond. Well, what we need is an enzyme, lactase, that can hydrolyze the beta bond into galactose and glucose, and then these are used in metabolism. If, however, we lack this enzyme, the lactose will continue to accumulate, and your body will try to digest it. And it will end up in your intestines where it is attempted to be digested by bacteria. And the question is, how does this feel to you? Cramping, GI distress. Again, it's what we would call uncomfortable, but it's not life-threatening. On the next page, the question is, what happens if we lack the enzyme to convert phenylalanine to tyrosine? Well, in the normal pathway, phenylalanine, in the presence of phenylalanine hydrolase, will be converted to tyrosine, and then it will go on to do something useful. It will be packaged into thyroid hormone, or it would be packaged into some other protein that is needed for the body. If someone lacks this enzyme, the phenylalanine will accumulate, and it has no choice but to go somewhere. Just like if Highway 43 is blocked, you have to just go somewhere else. In the presence of transaminase, the body will create phenylpyruvic acid. Now, I'm not sure if the benzene here strikes you in any way. After all, phenylalanine has a benzene, and so does tyrosine. But the reality of it is that phenylpyruvic acid is toxic to the brain, which means that if someone continues to consume phenylalanine who lacks this enzyme, he or she will continue to produce phenylpyruvic acid, toxic to the brain, and this leads to mental retardation. And this is why babies are screened for this at birth, because they cannot go very long consuming phenylalanine before mental retardation becomes irreversible. And we'll talk about this a little bit more in Unit 8. Enzyme inhibition. Why and when would we want to inhibit enzymes? Well, as we've seen before, we want to have control over our own enzymes. After all, you don't want trypsin and pepsin, digestive enzymes, to be on all the time. If they were, then that would cause those enzymes to basically auto-digest your own tissue. You also might want to control microbial enzymes. Just like in the last unit, we can try to control microbes as far as their proteins go by denaturing them with heat or organic solvents such as ethanol or isopropanol, or we might pressure sterilize them like we do for surgical instruments. So there are different kinds of enzyme inhibition mechanisms. And one is reversible competitive inhibition. Okay, so this one is the substrate and this one is the inhibitor. And what happens is you get reversible competitive inhibition when the substrate and the inhibitor are chemically similar and compete for the same site. So S and I are similar and compete for the same site. Okay? Now some examples of this are estrogen analogs. Okay, estrogen analogs. So these are things like birth control pills, certain plant-based substances like soy and isoflavones. Okay? And unfortunately, they also come in the form of pesticides that we typically get on fruits and vegetables. Okay? Now, the good news here is that this is reversible, and once the inhibitor diffuses away or is otherwise removed from the enzyme, then the normal substrate can bind and we get our products that we want and the reactions go forward. Another example is the normal pathway for DNA replication is in the form of part of a pathway that requires folic acid. Okay, so in the presence of folic acid, reactions can go forward that then yield processes that allow cells to divide. Okay? And that's because this process allows the formation of DNA and RNA that is needed for the nucleus, DNA needed for the nucleus for the cell to divide and replicate. Well, if you have this happening too much and you have a cancer or a tumor, one way to control the tumor is to stop cells from dividing too rapidly, which means stopping this process. Now, an analog of the folate derivative is methotrexate. Methotrexate, I'm putting too many R's in there. Methotrexate. So methotrexate is chemically similar to this molecule, which means that it binds the receptor so that this process cannot go forward. In our next segment, we will talk about non-competitive inhibitors that change the enzyme or the protein structure in yet a different way.